Hi, and welcome back to my channel, Learning Biology with Dr. Vanessa, where I take difficult biological concepts and make them easy to understand so you can feel confident mastering the science behind how your body works. In the last video, we saw how neurons fire electrical signals called action potentials down their axons. But to pass that signal on to another neuron, a muscle cell, or a gland, they must communicate at a site called a synapse. In many cases, this involves a small gap called a synaptic cleft that the signal must cross chemically. In other cases, like at electrical synapses, cells are directly linked by gap junctions, so ions flow straight from one to the other without a cleft at all. So how does that electrical signal cross the gap and continue the message? The answer lies in neurotransmitters, the nervous system's chemical messengers. Today, we'll explore how neurons communicate at the synapse, how signals can excite or inhibit the next cell, and how the message eventually stops so the system stays in control. As we have discussed in previous videos, the synapse is where communication occurs either between two neurons or between a neuron and an effector cell like a muscle fiber or gland. The neuron sending the nerve impulse or action potential towards the synapse is the presynaptic neuron, and the one receiving it is the postsynaptic cell. Most synapses connect an axon to a dendrite, axodendritic, but they can also connect to a cell body, axosomatic, or even another axon, axoaxonic. There are two main types of synapses, electrical and chemical, and they differ both structurally and functionally. Electrical synapses transmit signals directly from one neuron to another through tiny channels called gap junctions. These junctions connect the cytoplasm of adjacent neurons, allowing ions and small molecules to flow freely between them. Because the connection is direct, signals move almost instantaneously in milliseconds or less. This lightning-fast communication is perfect for situations where speed and synchronized activity matter, like reflexes, rhythmic breathing, and certain brain circuits. However, electrical synapses are less flexible because the message is essentially copied exactly from one cell to the next, leaving little room for modulation or fine-tuning. Most communication in the nervous system happens at chemical synapses, where neurons use neurotransmitters, those chemical messengers, to transmit signals across a gap called the synaptic cleft. Here's how it works. An action potential reaches the axon terminal, or the synaptic end bulb of the presynaptic neuron. This depolarization of the cell membrane causes voltage-gated calcium channels that are present in the synaptic end bulbs to open. Because calcium is more concentrated in the extracellular fluid, it rushes in. This sudden calcium influx triggers vesicles filled with neurotransmitters to undergo exocytosis, where the vesicles fuse with the membrane and release their contents into the synaptic cleft. The neurotransmitters then diffuse across the synaptic cleft and bind to receptors on the postsynaptic cell membrane. These receptors are often ligand-gated ion channels which open or close in response to the binding. When they open, ions move across the membrane, changing its voltage and creating a postsynaptic potential. If the binding of neurotransmitters opens sodium channels, the postsynaptic neuron depolarizes or becomes more positive. This is an excitatory postsynaptic potential, or EPSP, which brings the neuron closer to firing. If the binding of neurotransmitters opens potassium or chloride channels, the neuron hyperpolarizes or becomes more negative. This is an inhibitory postsynaptic potential, or IPSP, which makes firing less likely. If enough excitatory postsynaptic potentials add up to reach threshold, the postsynaptic neuron fires its own action potential, and the signal continues down the line. The key features of these chemical synapses are that they are slower than electrical synapses due to the time needed for neurotransmitter release, diffusion, and receptor binding, but they are highly modifiable and versatile, allowing for complex signal processing, learning, memory, and regulation. Once a neurotransmitter has done its job, it needs to be removed quickly, otherwise the neuron would stay activated indefinitely. The body has 3 ways to stop the signal. The first is reuptake. The presynaptic neuron takes the neurotransmitter back up for reuse, kind of like recycling. Sometimes nearby neuroglia can help with this process too, where it is called uptake. There's also enzymatic breakdown, where we can have specific enzymes in the synaptic cleft that destroy the neurotransmitters. For example, acetylcholine esterase breaks down acetylcholine in the synaptic cleft. And then there's diffusion. Some neurotransmitter molecules just simply drift away from the synapse. These processes ensure that the signal is quick, precise, and doesn't linger. Neurons rarely receive just one signal at a time. They're constantly bombarded by thousands of excitatory and inhibitory inputs. The neuron then adds them up in a process called summation. Spatial summation occurs when signals from multiple presynaptic neurons combine at once, where temporal summation occurs when one presynaptic neuron fires repeatedly in rapid succession. If the total depolarization at the axon hillock reaches the threshold, the neuron fires. If not, the signal stops there. Think of it like a competition. If enough excitatory signals outweigh the inhibitory ones, the excitatory signals win and the neuron fires. There are about 100 different substances that are known or suspected to be neurotransmitters. Neurotransmitters are the messengers that make these chemical synapses work. When they bind to receptors on the postsynaptic cell, they can either excite or inhibit that cell's activity. Neurotransmitters are crucial for everything the nervous system does. Every movement, memory, thought, and emotion depends on them. They can be divided into 2 classes based on size: small molecule neurotransmitters and neuropeptides. Some key small molecule neurotransmitters you should know: acetylcholine. This neurotransmitter excites skeletal muscle and plays a role in learning and memory. It can also be inhibitory and works to slow the heart when released by parasympathetic neurons. Glutamate. This is the main excitatory neurotransmitter found in the brain. It is essential for learning and memory. GABA and glycine. These are major inhibitory neurotransmitters that prevent overexcitation and maintain balance in the nervous system. Norepinephrine and epinephrine increase alertness and prepare the body for fight or flight. Dopamine is involved in reward, motivation, motor control, and mood regulation. Serotonin helps regulate mood, sleep, appetite, temperature regulation, and pain perception. It is also involved in emotional balance. Nitric oxide and carbon monoxide. These are unconventional gas neurotransmitters that diffuse directly through the membranes and help regulate blood flow, synaptic plasticity, and memory formation. Neuropeptides are a special class of signal molecules in the nervous system. Unlike classical neurotransmitters, which are usually small molecules that act quickly and locally, neuropeptides are larger chains of amino acids that often act more slowly and over long distances. They're made in the cell body of the neuron and then transported down the axon to vesicles in the synapse. Instead of simply turning the next neuron on or off, neuropeptides modulate neural activity. This means that they can fine-tune the strength, duration, or sensitivity of the signals, often working alongside those traditional neurotransmitters to shape how the nervous system responds. For example, they can make excitatory signals stronger, inhibitory signals more effective, or adjust how responsive a neuron is to repeated stimulation. Because of this modulatory role, neuropeptides are involved in complex and long-lasting processes like pain perception, stress responses, appetite, sleep, mood, and learning. Some well-known neuropeptides include substance P, which helps transmit pain signals in the spinal cord and brain, and the body's natural opioids, endorphins, and enkephalins, which reduce pain and produce feelings of euphoria. Others, like neuropeptide Y, help regulate appetite and stress. Understanding synaptic transmission is connected to medicine, behavior, and everyday life. Many common drugs and neurological conditions act directly on these signaling pathways. For example, SSRIs like Prozac work by blocking the reuptake of serotonin, keeping it in the synapse longer and helping relieve depression. Certain nerve gases and pesticides block acetylcholine esterase, causing dangerous overstimulation of muscles. Caffeine blocks adenosine receptors, reducing inhibition and making you feel more awake. Neurological disorders also highlight the importance of proper synaptic communication. In multiple sclerosis, the immune system damages myelin, slowing or disrupting the propagation of action potentials and leading to muscle weakness, coordination problems, and sensory issues. In epilepsy, uncontrolled bursts of excitatory activity can overwhelm inhibitory mechanisms, causing seizures. These examples show why understanding synaptic transmission is so important. Neuronal communication is finely tuned and can be easily disrupted, and knowing how it works helps us to see the connection between neuroscience, medicine, and everyday life. So that's the chemical side of neuron communication. Neurotransmitters carrying the signals across the synaptic cleft, excitatory and inhibitory inputs deciding whether the next neuron fires, and built-in mechanisms to stop the signal so that communication always stays controlled. With action potentials and synapses together, you now understand the core of how neurons talk, the foundation for everything else in the nervous system. If you enjoyed the content of this video, please make sure to subscribe, give this video a like, and turn on notifications so that you never miss out on new content. Thanks for watching, and I'll see you in the next video.